Plasma membrane structure and function. Today we'll be looking at the plasma membrane or cell membrane. So overview. The plasma membrane is the boundary that separates the living cell from its surroundings. Plasma membrane demonstrates something called selective permeability, allowing some substances to cross it more easily than others. Phospholipids are the most abundant lipid in the plasma membrane. Phospholipids are amphipathic molecules containing both hydrophobic and hydrophilic regions. The fluid mosaic model states that a membrane is a fluid structure with a mosaic of various proteins embedded in it. You better understand that by looking at the diagrams. So this first diagram shows the backbone of, or not really backbone, but the background of the molecules that form the membrane. So they are two phospholipids facing opposite each other. So there's two hydrophobic tail regions facing each other, and on either side there are the hydrophilic heads. The hydrophilic heads are facing the water, the hydrophobic tails are facing inward to each other. So you see those there, and then this is a zoomed in area of this showing the hydrophilic head region and then the hydrophobic tails region. So freeze fracture is a specialized preparation technique that splits a membrane along the middle of the phospholipid bilayer. Freeze fracture studies of the plasma membrane support the fluid's mosaic model, in which proteins are dispersed within the bilayer, with only the hydrophilic regions exposed to water. So here's an example of how here's the technique that in the picture of freeze fracture. So what you can see is that you freeze a cell in a cube in this case they used to use ice, but now I think they use materials that are like a polymer material. And they freeze it in it, and then they slowly split along. And when they split along, where the hardest region is, doesn't split, but the easiest part to split is between the layers of the cell. So between the cell membrane, both sides of the bilayer. So they have an extracellular layer and a cytoplasmic layer. The extracellular layer faces the outside of the cell. The cytoplasmic layer faces the inside of the cell. The results of this show that there are embedded proteins. So on the inside the cytoplasmic layer, you can see multiple embedded proteins. Inside the extracellular layer, you can see that there's not as many embedded proteins. And so what this gives us is this current model. So this is the phospholipid bilayer, and instead of just being a flat blanket of gray with on both sides with the yellow in the middle, what we see is that there are these proteins embedded, and these proteins have hydrophobic regions. The hydrophobic regions are found within the hydrophobic region of the phospholipid bilayer, the yellow tails. And then there's hydrophilic regions of the protein, and they are found on either end, and they stick out into the water. The fluidity of membranes. So phospholipids in the plasma membrane can move within the bilayer. Most lipids and some proteins drift laterally, floating through. Rarely does a molecule flip-flop transversely across the membrane. So we can see this diagram here. Lateral movement, as shown here, it's moving back and forth. Flip-flop, going up to down or down to up, very rarely. Fluid, membrane fluidity, we can see it very fluid when the tails are more spaced out, and there's more polyunsaturated tails. And we can see here where there's saturated tails, where there's fewer kinks in it. So it's much more viscous, much more tightly packed, closer to solid. And then we can see that in this bottom one, there are cholesterol molecules that have been added, and they increase membrane fluidity, as we'll see next, or later. How do we know that it's a fluid, and how do we know that the proteins are embedded in it but able to float around? Well, actually, there's a really interesting study that was carried out by scientists in which they took a mouse cell and a human cell. So mouse cells and human cells, as you can imagine, are slightly different, and they have different membrane proteins on the surface of them. So on the surface of the mouse cell, there are mouse membrane proteins, and on the surface of the human cell, there are human membrane proteins. They created a hybrid cell. They made the cells bind together. They used a little bit of electricity. They caused the cells to do something that's not normal. But what they found was that after an hour, all the proteins were intermixed. There wasn't a section of mouse cell proteins and a section of human cell proteins like there was in the beginning of the hybrid cell. In the end, all the proteins were mixed. So membrane fluidity. As temperatures cool, membranes switch from a fluid state to a solid state. So this is important to remember. Your normal membrane, the membrane that's in your cells, is actually a fluid. So it's a liquid that is very thinly enclosing your cell. The temperature at which a membrane solidifies depends on the type of lipids. So membranes that are rich in unsaturated fatty acids are more fluid than those rich in saturated fatty acids, as shown in the diagram that we saw. But membranes must be fluid to work properly. They are usually about as fluid as salad oil. The steroid cholesterol has different effects on membrane fluidity at different temperatures. At warm temperatures, such as 37 degrees Celsius or human body temperature, cholesterol restrains movement of phospholipids, so it slows it down from becoming too fluid. At cool temperatures, it maintains fluidity by preventing tight packing, so it prevents it from becoming solid. So steroid cholesterol being embedded in the membrane is very important. Membrane proteins and their functions. A membrane is a collage of different proteins embedded in the fluid matrix of the bilayer. Proteins determine most of the membrane's specific functions. Here is a diagram of the actual membrane and all of the things that are involved in it. And so you can see, we can label the detailed structure of this is for an animal cell's plasma membrane. So we see the phospholipid bilayer. We see cholesterol embedded in it. So you can see the cholesterol embedded there, the phospholipid bilayer in the background in the yellow and the gray. You can see that there are proteins that are embedded. These are called the integral proteins. There are proteins that are on the interior cytoplasmic side. These are called peripheral proteins. There are microfilaments of cytoskeleton that attach to the peripheral proteins. So these are part of the cytoskeleton of the cell. This is all on the cellular side. And then on the extracellular side, we see fibers of the extracellular matrix. And many of those fibers connect to glycolipids and glycoproteins. If it's a glycolipid, it's attaching directly into the phospholipid bilayer. If it's a glycoprotein, it's attaching a carbohydrate chain to a protein. So glyco means carbohydrate, and the green here are all carbohydrates, and they, in some cases, are attaching to the extracellular matrix. Peripheral proteins are bound to the surface of the membrane. Integral proteins penetrate the hydrophobic core. Integral proteins that span the membrane are called transmembrane proteins. The hydrophobic regions of an integral protein consist of one or more stretches of nonpolar amino acids, often coiled into alpha helices. So here is a model diagram showing the structure of a transmembrane protein. So you can see that there are regions that are hydrophobic and there are regions that are hydrophilic. So the hydrophobic regions would be here in the middle, and the hydrophilic regions would be here on top and here on the bottom. So both cytoplasmic and extracellular side would be hydrophilic. All right, the 6 major functions of membrane proteins. So transport, enzymatic activity, signal transduction, cell-cell recognition, intracellular joining, and attachment to the cytoskeleton extracellular matrix. So let's break those down a little bit. So some of the functions of membrane proteins. So here are the diagrams that sort of show each of these processes in action. So you can see transport, which we're going to look at much more in depth. Enzymatic activity, which we're going to see some detailed examples when we get to cellular respiration. Single transduction, which we don't really cover in this course. Cell-cell recognition as well, intracellular joining, and attachment to the cytoskeleton and extracellular matrix. So really just the first 2 we're going to look at more in depth. The role of membrane carbohydrates in cell-cell recognition. Cells recognize each other by binding to surface molecules, often carbohydrates, on the plasma membrane. Membrane carbohydrates may be covalently bonded to lipids, forming glycolipids, or more commonly to proteins, forming glycoproteins. Carbohydrates on the external side of the plasma membrane vary among species, individuals, and even cell types in an individual. And this is part of the immune system, something that we don't really cover in this course. Membranes have distinct inside and outside faces. The asymmetrical distribution of proteins, lipids, and associated carbohydrates in the plasma membrane is determined when the membrane is built by the endoplasmic reticulum and the Golgi apparatus. So as you can see, there's a sidedness to the finished product, and that is all because of the way that in this diagram, you can see that the transmembrane proteins are created and sided into the membrane. And so when they go from the rough endoplasmic reticulum to the vesicles and then to the Golgi apparatus, as you can see, then through the Golgi apparatus, they leave with their glycoproteins attached or glycolipids attached, depending, they have the carbohydrates added to them, and then they go out and form into the cell. And you can see in some cases they also secrete a protein separately, and they are what forms the membrane glycolipids along this surface. A cell must exchange materials with its surroundings, a process controlled by the plasma membrane. Plasma membranes are selectively permeable, regulating the cell's molecular traffic. Hydrophobic or nonpolar molecules such as hydrocarbons can dissolve the lipid bilayer and pass through the membranes rapidly. Polar molecules such as sugar do not cross the membrane easily. In the next video, we'll look at cell transport.